next we'll move on to our next segment. So earlier we had the opportunity to hear Randall discuss the inverse problem. Now we'll transition to a topic at sort of the core of his work. So in this talk, he'll walk us through the challenges and possibilities of whole brain emulation. Please join us in giving a warm return to Randall Koene.
So I spent a lot of my time, or a bit of my time, in my earlier talk indicating something we dubbed the meta problem in brain emulation. And I listed a number of ways to move forward, one of which was to use in silico generation of example data as a testing ground for methods and explorations regarding scale separation. The following is an introduction to an open source, open science platform we've built here in our research group at Carboncopies. And I'll be focusing mostly on the reasons why. In other words, the ideas behind it, the choices we made so far. So the talk will be theory and method today, and at the next workshop, which is probably in May, the focus will be practical demonstrations and hands-on experience. This talk's got two parts. The first part is about the platform, and the second part is about the challenge to the community based on that platform. All right, so the main purpose here is to provide a platform for the systematic generation of examples that can be used to address the deep meta problem of brain emulation. There is the fundamental problem of identifying and specifying subdomains of research that have a particular purpose in mind with regard to brain emulation, and of maintaining mental clarity within that subdomain about corresponding success criteria. It's a word that I keep bringing up again. In my talk earlier, I tried to express a couple of axioms that affect exploration once you find yourself in such a subdomain, once you've expressed your purpose and goal. There were the two axioms. Axiom 1, the emulation efficiently retains necessary function. The emulation has a footprint that is as light as possible, and the emulation has abstracted as much of the original substrate as possible. And then the second one, robustness requirements demand scale separation, and this must be true for brains. We can ask ourselves, how do we ensure that emulation attempts are operating in accordance with these axioms within the particular context of a purpose, so a set of success criteria? And we can look at the practical difficulties that arise, which is where we hope that our proposed approach can be helpful. I'll say in advance that where I describe examples and scenarios in the rest of this introduction, I'm coming from my own interest, which is primarily in the application of brain emulation to the domain of medical technology, quite close to some of the possibilities that Razvan has mentioned before. So mind uploading is definitely one of them, from simple neuroprosthetics to whole brain neuroprosthetic, which I guess is then mind uploading. Nevertheless, I hope that it won't be too hard to see how the same tools can be useful in an adjacent subdomain. Let's say you're attempting a functionalization that is more extensive or more sophisticated than the ones already published and presented today. And let's say something doesn't appear to work as intended. How do you discern why and what to do about it? If you're exploring a method of analysis and functionalization, for example, in Drosophila, doesn't the utility of synthetic examples automatically arise? See, for example, the way in which the work by Jan Lappalainen and their team presented, as he presented, but where I then asked him to talk about the synthetic networks they generated to explore the strengths and weaknesses of the approach. In that case, they were looking at the effect of sparseness. So a simple way to look at that is to say, hey, can your approach discover what we've hidden in this data? If it can, then that increases my confidence that success criteria may be met when applying those methods to real data. Yeah, I should have popped that up earlier. This is Lappalainen's thing, of course, here. This is the example that he also showed when I asked him about that. So this is where they trained multiple networks to test differences as you have more or less sparsity in the system. Using synthetic data to augment or substitute for real data at some stage of a modeling project is not new. I thought it might be useful to list 6 examples. The first one's already up. There's a more complete list in the literature references of our upcoming paper draft about the approach, where we compare with a larger variety of domains and explicitly compare how we position the use of synthetic data with the applications in those other examples. So this first one, ArtImagen, or the Artificial SEM Image Generator, SEM images of various kinds, including gold on carbon and some semiconductor structures generated, and they also simulate numerous artifacts such as noise, drift distortion, edge effects, and that synthetic data is then used to assess imaging, metrology, and other techniques. They emphasize how this allows them to carefully specify the degree to which artifacts appear. In leveraging generative adversarial networks to create realistic scanning transmission electron microscopy images, synthetic data is used specifically to improve automation and machine learning in EM for autonomous data collection and processing. They highlight how such data sets can improve generalization to large data sets under varying experimental conditions. Our goals are, of course, different than these examples in that we're aiming to support methods and algorithms development, producing working connectome-constrained models of neural circuits on top of segmentation and identification. In generative model of the connectome with dynamic axon growth, synthetic data is meant to explore insights into the wiring principles underpinning brain network organization. The authors explain that while earlier studies could approximate numerous statistical properties of empirical networks, they typically fail to explicitly characterize an important contributor to brain organization, namely axonal growth. By including chemoaffinity guided axonal growth, their synthetic data generates axonal fiber bundles with brain-like geometry and features of complex network architecture consistent with the human brain. Further, they explore connectome dimensionality reduction and comparison of parameters between groups. It's worth noting that our recently updated version of NetMorph, which I'll talk more about in a bit, also uses both stochastic phenomenological algorithms to produce cell type specific morphologies, and chemoattraction is indeed also included as a means to direct the production of useful connectome reservoirs. You'll see in a bit why that matters. In generating brain-wide connectome using synthetic axonal morphologies, synthetic data is used to study the relationship between healthy and pathological local and long-range connectivity, spanning scales usually addressed by either EM or MRI, i.e. both projectome and connectome. And in generative models for large-scale simulations of connectome development, the authors specifically point out the problem that knowledge of the ground truth topology of the connectome is lacking, which stands in the way of exploring processes at scale regarding development, aging, and neuropathologies. Biologically meaningful simulations using synthetic graphs with controllable parameters can complement real data analysis and provide critical insights. Generative models can be highly valuable tools for creating large data sets of synthetic graphs with known characteristics. And finally, in medical image synthesis for data augmentation and anonymization using generative adversarial networks, synthetic MRI is used to balance medical image data sets where pathological findings are generally rare. Synthetic images with brain tumors are generated using generative adversarial networks, or GANs. The data set is then applied to segmentation learning. There are also quite a few platforms that use synthesized models and synthetic data generation to explore interactions at the molecular level, where this approach has been explored and accepted to a large degree. You can find many examples at the eBrains modeling and simulation software page. So the loop shown here should look fairly familiar because it's the scientific process, and it's an expression of the meta problem I mentioned in the earlier talk. You collect some data, so you have observations about the world, or in our case, about a brain. You have some hypotheses that are based on those, and you carry out analysis and interpretation, which leads to a model for a specific piece of brain tissue. Here shown in 2 steps that are typical when working primarily with image data. First, finding system structure and identifying components based on prior knowledge, then using hypotheses and prior knowledge to determine parameters for selected component models. Rounding that out with a process of estimating remaining parameters, approximating at what is hopefully a useful level at which to solve black box problems. Given the purpose of the overall system model, in other words, your success criteria, here, this is labeled translation, because it's a translation of collected data constrained by structure and prior knowledge. Then you have to verify that your model is working as intended, meaning that it represents the hypotheses you intend to test. And of course, you need to validate the model using validation metrics that are meant to address a set of success criteria, which can and should appear at multiple levels, satisfying constraints at the component or subcircuit level. But most importantly, at the system level, where you attempted to specify your purpose or goals in terms of success criteria. Sometimes this means using the model to predict circuit responses or system behavior. Often you may be interested in the behavior of some subsystem, while the remaining network is a potentially confounding context. Conrad already mentioned that when he showed his little y equals something about x and then z plus epsilon. The x and z there, the part that you care about is the x and the z was the part you don't really want. If the experiment was well designed, then you hope to quantify how well the model reflects reality, again within the context of your purpose, and that errors will point to possible corrections which can qualify the hypothesis you had going in. Then the process begins to iterate. This is part over here. Because your insights can either lead you to adopt a very different approach or different hypothesis, or it might feed back into modifications, improvements of various kinds. Generative meta-analysis proposes a systematic protocol that aims to accelerate the necessary improvement loop for analysis methods that are applied to the discovery of valuable subsystems, meaning subsystems that satisfy success criteria within large, complex nonlinear systems. To do so, by supporting synthetic ground truth-based testing and discoveries in scale separation. Testing of analysis methods is carried out by generating many example systems in silico, ensuring a fully known ground truth of a sort that may otherwise be unobtainable. Examples are generated at multiple degrees of difficulty, and variables included can be controlled precisely. All right. The design of the platform isn't strictly limited to neuroscience, although that is primarily our interest. Okay. In the most general sense, you're dealing with a system that contains a large number of interacting nonlinear components, shown here as this wild network in the background. In the most general sense, you're dealing with a system that contains a large number, I'm repeating myself. That system contains something, I'm getting tired, I think. That system contains something that's of interest or of value. That's shown here in green. For example, a subsystem that sustains working memory or that encodes patterns as attractors, or one that carries out motion detection. You want to carry out system identification in a way that satisfies success criteria. The actual data you have may come from a larger system, for example, from a volume of neocortex or tissue in the visual area. So you could say that the subsystem you care about is is obscured, hidden, and affected by the larger system in structure and function. All of that is further embedded in a substrate, one that, using your current hypotheses, can be considered passive with regard to the operations you're investigating, even though that passive environment may very well have some effect on the signals you mean to include. That environment is represented here by the colored hexagons, and the fact that they're different colors is meant to indicate that the passive environment can be composed in a heterogeneous way. For example, cerebrospinal fluid, vasculature, glia, and so forth. To test your methods of system identification, reconstruction, functionalization, you want to apply validation metrics to suit your success criteria. But while you're still working on those methods and trying to carry out rapid development, wanting to see how capable they actually are, it's either not easily possible or far too costly to do all of that by building the technology needed to collect sufficient real-world ground truth data, especially if your methods exploration, so the different methods you're trying to explore and compare, would involve different types of technology or their modification and extension. For example, you wouldn't want to set up high throughput data collection platforms of multiple kinds in neuroscience, or if this was a problem in physics, you wouldn't want to build multiple test particle accelerators. The studies you're carrying out are also meant to explore potential hypotheses before committing to them. You want to learn about functional constraints and corresponding instances of scale separation to help support or discard ideas about which components at some resolution should be considered part of the nonlinear system relevant to your goals, how you should model those compartments and their interactions, and what data you're going to need. In summary, reasons to want an in silico platform for these explorations are to compare different methods and determine which are best in what circumstances, when, how, and why they break. And to figure that out, you want to be able to easily move between simple, unconfounded small examples to more complex, more confounding, larger examples, with careful control of each factor involved. You may want to run many experiments at once and iterate easily, while producing studies where the results are based on a God's eye view. Ultimately, studies like those improve clarity and reduce risks while building simulations, data collection technology, and methods. The platform for this generates synthetic examples with a fully known ground truth. While systems of unspecified function are possible, the application we highlight here purposely hides a so-called meaningful subsystem within a larger one to emphasize problems involving prioritization of overall success criteria, as opposed to optimization of component fitting, scale separation, and constraints of neural coding. Obviously, evaluation has full access not just to the reverse engineered or emulated model produced by methods being tested, but also to the synthetic ground truth. And functional results can reflect both the situation without using prior knowledge, for example, by using random stimuli, as well as results that are based on stimuli that specifically target the operations of the hidden system. For each generated example, the platform can carry out a battery of virtualized data collection. At the moment, our platform focuses on collecting synthetic stacks of electron microscope images, virtual calcium imaging, and virtualized electrophysiology. For example, in one run, the platform can generate n synthetic ground truth examples specified for m different combinations of factors such as cell types, number of cells, density, signals included, noise or artifacts in the collected data, and evaluate the resulting emulations achieved with p different approaches. If carried out as part of our challenge, studies are double-blind. The red network shown here is just for illustrative purposes and is meant to represent a reconstruction and emulation of the green subsystem in the example. You can see that this is a reconstruction in something else because it's got straight lines instead of curves. A collection of examples can be generated as a way to explore almost one variable at a time, effects along multiple dimensions or axes of interest. This is shown here as if in a control panel, where you can vary between small and large example systems, systems with fairly simple architecture or more complex, virtual data collection without noise or various forms of noise and artifacts, a bare system without confounding surroundings, or an embedded system. Exclusion or inclusion of components like gap junctions, synapses with one or multiple neurotransmitter types, systems containing rhythmic modulation, details of perineuronal nets, or examples with a meaningful hidden function versus random networks, example models at low resolution, such as point neurons, or detailed at higher resolutions, and output data at low to high resolution, etc. And then there's automation for generating data collecting from a batch and exploration for ranges of these configurations, and evaluation and comparison of the results achieved for methods being tested and contrasted here, depicted here for just 3 configurations. So we've got this 3-dimensional little exploration space. Generative meta-analysis is intended to be a development tool that helps pretest analysis methods more easily and more rigorously before the best performing ones are applied to biological data where the ground truth is rarely fully understood. Iteratively applying the protocol should lead to improved analysis methods, such as are required for functionalization, an improved understanding of the success criteria for brain emulation, an improved understanding of scale separation in the brain's neuronal circuits and mechanisms, and improved synthetic example generation for further meta-analysis as the insights are fed back into updates of the platform itself. Using this protocol, generating many examples containing multiple types of neural circuits subject to realistic constraints at multiple scales and levels of complexity with different degrees of noise acts as a means of validating analysis methods that are proposed. So the question is, can a proposed method of analysis deliver insights constrained by neural coding at appropriate hierarchies of scale separation? A possible analogy for this type of performance testing is passing a flight simulator exam. In this analogy, the methods being tested are the cadet pilot in training. After hundreds of hours demonstrating skill in the simulator, confidence in the pilot's skills reach a level where she's likely to succeed and successfully handle real-world conditions. Otherwise, you wouldn't let them into a plane. We hope that this level of exploratory control will help determine the significance of constraints imposed by regional neural coding protocols. One of the obvious examples where system constraints impact scale separation is through morphological structure, as expressed in terms of the many tomes, the connectome, but also the synaptome, the complete set of synapses in the nervous system, often mapped at high resolution, the transcriptome, the full set of RNA transcripts produced by the genome in a specific cell or tissue, the dynome, the dynamic states of a system, often used in relation to brain activity, our cellular function, the pathome, the set of pathological changes in a biological system, and the metabolome, the full set of metabolites present in a biological sample. Knowledge of the neural coding strategy used in meaningful communication and processing within a specific subcircuit can provide information about functional constraints that are imposed and that can simplify modeling. For example, specific neurons may communicate through bursts of action potentials via firing rates above some threshold rate, or may synchronize and elicit synaptic modification in a manner that's highly dependent on pre- and post-synaptic spike times, so the spike intervals. Or groups of neurons may be connected such that they form attractors in terms of cued pattern reactivation, and in some cases, such as circuits involving auditory sound localization, the onset time of postsynaptic activity at spatially separated synapses on the same dendrite may be crucial to dendritic computation. Examples of constraints that go along with coding strategies mentioned up there are noise thresholds established by signal-to-noise ratio, the necessary separation of synchronized patterns of activity by recurrent inhibition, firing synchronization modulated by rhythmic input and corresponding phase locking of activity, or the activity of interacting regions may be dominated by strict phase offsets between two communicating regions, for example, to accomplish encoding in one region of entorhinal cortex based on retrieval from another region in entorhinal cortex, or guaranteed cued recall of full patterns of activity within attractors with a minimum cue of a certain size, or pattern-dependent activity within neural populations. And of course, the model architecture itself is connectome constrained, as was demonstrated in Jan's talk, for instance. In the first step, so I'm going to go through the protocol now. And the first step of the protocol that we use the neural outgrowth simulator. I should click on it so that, because it takes a little while to go through. The neural outgrowth simulator netmorph that I published in Neuroinformatics back in 2009, we extended it significantly to embed it into the platform and for the needs of the protocol. It's configured to produce a specific architecture or a specified architecture in terms of regions, layers, cell types for which outgrowth is simulated until the detailed morphology of resulting neurons and their networks satisfies the needs for a reservoir with sufficient candidate synaptic connections to express a meaningful neural circuit. And the clip that I just showed was a time lapse of one example of such growth. Oh, and then here we have that issue. There we go. In step 2, pruning and tuning, in the second phase, pruning and tuning takes place as stimuli are presented to the connectome reservoir so that it does indeed express a desirable neural circuit function. For example, autoassociative memory with attractor dynamics using excitatory and inhibitory recurrent connections. And the example shown here is a very small example. It has 4 layers and not that many neurons, but you can already get the gist of what is produced here. And the third step, embedding. So the subsystem that we just generated lies hidden within an encompassing nonlinear dynamic system, meaning neuronal circuitry that doesn't directly relate to the function. Again, I'm referring back here to what Conrad was already putting into one of his equations there when he was trying to do the inverse problem, or for a system where the subcircuit that you have that you're looking at is actually surrounded by many more neurons. So it's not just the one that you've been recording. In a third phase, the netmorph output is embedded within a simulator of naturalistic passive components that don't directly contribute but may affect signals, for example, extracellular materials. And that embedding is done by a component of the platform called BrainGenix NES, as well as meshing and voxelization of cells, scaffolding the cells with dynamic properties, and setting up virtual data collection devices such as a configuration of high-throughput virtual electron microscopy, virtual calcium imaging, and a specified arrangement of virtual neuropixel-like probes for electrophysiology. In a fourth phase, virtual data collection is carried out in that example system. This creates synthetic data for, the electron microscopy, calcium imaging, and electrophysiology. The resulting data stacks are available to the analysis method being tested without providing direct knowledge of the underlying ground truth of the example system or its embedded meaningful function. For experimentation, we give the data together with any important physical specifications where those intentionally deviate from reality. For example, it's important to know if you should find visible postsynaptic densities in the images that you're looking at, or if the cells grown match morphological specifications of real cells, such as pyramidal cells, stellate cells, etc., or if a simplified representation was used. So experimenters receive the data stack generated by virtual data collection from the virtual tissue. They don't receive direct information about the synthesized ground truth behind it. The analysis methods being tested make a best effort at system identification, for example, by a process of model initialization constrained by an identified connectome, followed by a process of functionalization, and thereby produce a proposed resulting emulation. Keep in mind that at this stage, the experimenter can do anything they like with the data. So really what I was showing here is just an example. It could be anything. They just get the data and they deliver what we're calling the emulation. So it could be what Phil did and what Jan did, but it could be something else. The resulting model, its structure and dynamic responses are used by our platform API to carry out the ensuing comparison with the example ground truth. The quality of the emulation is validated using direct knowledge of the example ground truth, comparing the structure that was discovered, as well as the correctness of function. The evaluation points out strengths and weaknesses in the performance of competing analysis methods, which can be scored and compared across a wide range of approaches. A quick sip here. It's entirely possible to apply the same protocol using randomly generated examples, but here we highlight evaluating analysis methods for their ability to correctly discover a meaningful subsystem, because it's an explicit effort to aim the evaluation not towards optimizing performance on the reconstruction and reverse engineering of component-wise details, but instead towards goal-directed success criteria that are intended to emphasize cognitive experience and to discover points of scale separation most relevant to those. As I mentioned, I'm most interested in the medical applications. Currently, all of the steps that I just mentioned have been implemented in at least to some degree. The framework has been completed. We're still improving each part to get what we consider our prototype or MVP, demonstrated by something like generating an EM data stack that is a simplified version analogous to Drosophila Visual System EM data, and demonstrating that something like the connectome constrained reconstructions that Jan talked about can be tested on that data. We're expecting and hoping to successfully run that prototype test by May, which is why we anticipate scheduling the second workshop in this series then, to offer hands-on demonstrations where participants can play with the protocol. An important aspect of the development between now and then will be to improve the interface by adding sufficient automation, so that an experimenter can specify exploration ranges rather than having to tinker directly with the configuration. We want an experimenter to be able to set up a batch of experiments and then receive detailed results organized by experiment with comparative evaluations tabulated. If the experimenter needs to work manually with the data to produce a result, perhaps because they rely on a batch of separate scripts or something like that, then that step of the protocol takes place manually. But hopefully, the experimenter will have learned to take full advantage of our API and can feed the data directly into their method of model building from EM data plus physical knowledge and estimation techniques. If they can do that, that would be the most efficient way to make use of the platform as a source of synthetic data, an automated way to explore multiple circumstances, and an automated way to evaluate results of the attempted emulations. If you're interested in details of a small worked out example, I suggest watching our recorded update of work on the platform that's available on our media server at videos.carboncopies.org, or better yet, join us at the next workshop to fiddle with the controls. And with that, we get to the second part, which is shorter than the previous one. The brain emulation challenge. So how far can we get using currently available data? That would be a really good thing to figure out. Let's see what everyone can do. The challenge is a complement to the protocol itself. It's meant to provide an equal playing field for different and competing ideas, and is meant to ease the point of entry for more people within academia and beyond to get involved with the problems of system identification, functionalization, and brain emulation. We hope that providing a space where approaches can be tested and contrasted raises motivation to address the relatively underserved areas of problem specification in terms of goals, criteria, and metrics, and methods development and rigorous testing. There we go. So our code is all available, and it's going to be supported by published documentation and theory. You can also get access to the running system on our server nodes, because for most people, this is much, much too large to run on a small system that you might have at home. Our aim is to help accelerate the work of all parties. We're fully welcoming to collaborators, partnerships, and forking of our system to try alternatives. It's also entirely possible to simply participate in the challenge without participating in platform development. We're grateful for constructive critique and recommendations that help direct further improvement. This is a great time to get involved, because right now we're very open to suggestions that can improve how well our platform suits the goals of your research, specify formats and characteristics of the data you need, or the evaluation output that's most helpful. In other words, consider this a potential resource, a way to outsource some aspects of your work with a very receptive team on the other end. As soon as we've finished prototype testing, and probably at or immediately following the hands-on workshop, we aim to launch the very first version of the challenge. At this point, we aim to submit preprints that explain and describe ideas and purpose and the technology behind the platform and the challenge. The domain brainemulationchallenge.org will host documentation and worked out examples. Okay, let's look at the protocol itself. So here, boxes in yellow are things that a participant does. So there's going to be an entry point here to take part in the challenge once that brainemulationchallenge.org site is open. Doing so will involve completing a registration form and picking the type of challenge examples that are interesting and suitable to the participating group or person. You will then receive access to 2 sets of synthetic data, information about success criteria, and the evaluation that will be applied, as well as relevant physical assumptions that intentionally deviate from reality. One set is generated specifically for you, but standardized to achieve a difficulty level. This is meant to reduce the chance that participants benefit heavily from results previously published by other participants, even though learning from prior results is clearly possible and is, of course, intended to facilitate progress. So the other set is meant for direct comparison with results produced by other participants or your own previous entries to the challenge, and is standardized to be the same among participants who choose the same problem space. You then follow the protocol that was already outlined in earlier slides by subjecting the synthetic data to your chosen approach or approaches, and you use the platform API to submit the results in the form of structure and dynamic output in the form that suits your problem choices. For example, in the form of spike trains or cell membrane potential. You will have the option to provide additional data, such as complete models. You receive evaluation reports and scores. For challenge purposes, we'll be limiting the types of data that we're evaluating, in part because success criteria are paramount and not necessarily whether a method chooses to identify ion channels or uses larger black boxes to approximate. We'll also be available to help participants work with the data and the API to respond to feedback about important matters such as format, protocol, or aims of challenges, or the way that success criteria are chosen, specified, and evaluated. We do hope to track a leaderboard of sorts, although the initial version of the challenge, at that stage, we expect there will be quite a bit of learning and improvement. And then eventually in 2026, we hope, in collaboration with our sponsors, to make prizes available as an added incentive. As mentioned in my earlier talk, there are certainly risks when using in silico examples. The primary 2 risks are as follows, and you may have already heard a bit of this. The first one is misdirected performance optimization. If a specified set of success criteria is insufficient, or if emergent criteria are not sufficiently tested by corresponding validation metrics, then offering a challenge and performance scores will lead to competing methods or algorithms optimizing performance in a manner that can be misleading. We're aware of this and will be watching those criteria carefully. And the second one is drawing conclusions on what I'm calling alien ground truth. Any simulated ground truth carries within it the risk that assumptions used to generate the ground truth system and its corresponding structural and functional virtual recorded data contain errors. When a fully known or fully annotated real-world ground truth is unavailable or unfeasible, that's unavoidable. In R&D, this is addressed by iterating the closed-loop development process in a manner where both the analysis method being tested and the ground truth simulation platform are improved together. Ideally, this is done by testing the best-performing analysis methods on real-world data in order to introduce previously unknown or unrecognized corrections. The risk is not unique to our situation, not only does it apply to all use of synthetic data, it's also implicitly contained in decisions about what type of data to collect and the limitations of those techniques, as well as decisions made in attempts to reconstruct. So we can't make up the truth, but we can provide a low-cost, rapid way to test how well approach X handles condition Y. Since we want the advantages of rapid turnaround time, low resource cost, and systematic targeted methods exploration, we have to be acutely aware of and deal with the associated risks. Previously mentioned examples of synthetic data applications and the effect that challenges have had on the development of AI give plenty of evidence of the value. So, how can you engage with this challenge? As a challenge participant, I encourage you to join our next workshop to get a feel for the platform, the experimental protocol, and the challenge protocol. Then please sign up for the challenge as soon as brainemulationchallenge.org opens up. And please give us every bit of feedback that you care to give. There's no need to feel stuck, confused, or irritated, because we know that the scientific process I described earlier is one where all parts have to improve in a fully connected cycle. And that, of course, includes our methodology, synthetic example data, interfaces, and evaluation. As a platform and challenge development partner, well, remember that we're an open science nonprofit. Our only goal, is to help advance the field wherever we can do best. We're not trying to profit from your work or to compete with your expertise. We want to be a resource and a community, much like preprint services are a resource and a forum. We highly encourage anyone who wants to help make this the best it can be to work directly with us. And of course, if you want, you can take it in another direction if you want. But even then, if you're forking it, then we hope that you operate in the same spirit of cooperation. As challenge funders, well, as many of you are probably aware, having had to find funding for your own work, it's not easy in this domain, especially as an academic or a nonprofit. The reasons are many, and they probably deserve a discussion all by themselves. As it stands, we're operating on a shoestring budget. If you'd like to help us provide resources and increase awareness, which we try to do as leverage points with accelerated returns, we strongly encourage you to help fund our efforts by donating to the Carboncopies Foundation or by reaching out to us about partnerships and other forms of support. In conclusion, as you know, this is the first part in a two-part series. I've described the idea of generative meta-analysis and its connection with the process of methods discovery and creation, as well as the entry point for synthetic data as a workaround to the unavailability, high cost in resources or time when it comes to the need for ground truth-based data in the process of testing and validation. I pointed out that there are risks associated with over-reliance on synthetic examples and performance-based scoring, which one must always remain vigilant of to understand carefully how best to use explorations with synthetic data as a systematic means by which to identify strengths and weaknesses of assumptions, analysis, and interpretation. I gave an introduction to the protocol we build our platform around and how you can make use of it. And finally, I introduced the Brain Emulation Challenge and the open science approach we aim to leverage through that. I hope this has made you ready and eager to explore as soon as brainemulationchallenge.org launches, and that you want to help build excitement and motivation to pull in more people and more resources into this problem space as quickly as possible. And right here on my thank you slide, my final slide, you can see that we're also directly looking for help in certain areas. Thank you so much.